Alright guys, this video is over the cell cycle. We're going to talk about mitosis and cell division. So this one's kind of a long one. Make sure you're taking your C notes. We're going to go over them in class. Make sure you write down any questions that you might have. Alright, so let's get started. So our objectives today are to describe the stages of the cell cycle, including DNA replication and mitosis. We're going to describe the importance of the cell cycle for growth of organisms. Alright, so first we are going to define the cell cycle as the sequence of cell growth and division that occurs in a cell between the beginning of one cell division and the beginning of the next cell division. This cycle includes preparation for the cell division and the actual process of cell division. So here's a picture here. We're going to go through each one of these stages. So here we go. There are two major stages of the cell cycle. The mitotic phase is when the cell actually divides, while interphase is the cell in the preparation parts for division. The mitotic phase includes mitosis and cytokinesis. Mitosis is the process of nuclear division, and cytokinesis is the division of the cytoplasm. While cytokinesis is not part of mitosis, it occurs at the end of the mitotic phase. So in this diagram, the letter M is referring to the mitotic phase. G1, S, and G2 are stages that occur during interphase. Okay, so G1 is where the cell begins and the cell grows. Here is S, we're going into DNA replication. G2 is where the cell prepares to divide, and then mitosis is where it's actually going to divide. So G1 phase of interphase is the primary growth phase of the cell. The cell grows in size. Cells often pause in G1 before the S phase of DNA replication. They remain in this state for hours to years before resuming cell division. Some, such as muscles and nerve cells, remain there permanently. On the diagram, this state of rest is signified by the letter R. If and when the cell cycle resumes, the S phase occurs. The S represents synthesis of genetic material, known as the process of DNA replication. Once the DNA has been replicated, the cell prepares for the cell to divide for the second period of growth called the G2 phase. Throughout the phases of interphase, organelles such as mitochondria replicate as well. A somatic cell is a body cell in a multicellular eukaryotic organism. All of the cells that compose a particular eukaryotic organism are considered somatic cells except for the sex cells or the gametes. Somatic cell in a particular organism contains the same number of chromosomes. For example, the human somatic cell, whether it be skin or muscle, contains 46 chromosomes. So mitosis is the process by which the daughter cell receives an exact copy of chromosomes present in the parent cell, and then it forms two identical daughter cells. Mitosis ensures that new somatic cells receive the correct number of chromosomes. Alright, there's 4 phases in mitosis. There's prophase, metaphase, anaphase, and telophase. Alright, prophase is the first phase of mitosis. Remember that this follows interphase, in which DNA has already replicated. The chromatin, the hereditary material consisting of DNA and protein, begins to coil up into short, thick rods, and we call them chromatids. A chromosome is a unit of coiled chromatid. Because DNA is duplicated, the chromosome contains 2 of these chromatids. On the chromosome diagram, each half composes 1 chromatid. These 2 chromatids are identical and often called sister chromatids. A centromere is at the point at which the 2 chromatids are held together. So if you look right here, this right here are the identical chromatids, and right here in the middle is the centromere. During prophase, two pairs of centrioles appear to one side of the nucleus, outside of the nuclear membrane. The two pairs of centrioles move apart and to opposite sides of the cells. Spindle fibers form between the two pairs. Note that plant cells develop spindle fibers but lack centrioles. This diagram represents prophase within an animal cell. Okay, so here's our centrioles and here's our spindle fibers. The nuclear membrane eventually breaks down and disappears, and the centriole pairs reach opposite sides of the cell. At the end of prophase, chromosomes become attached to centrioles by the spindle fibers. Metaphase is the second phase of mitosis. Chromosomes are pushed and pulled by spindle fibers toward the middle of the cell. Eventually, the chromosomes are arranged along the cell's middle, also called a metaphase plate or spindle equator. The centromere of each chromosome becomes attached to a separate spindle fiber. Anaphase is the third phase of mitosis. Anaphase begins with the separation of chromatids at the centromere as the spindle fibers shorten and pull. Remember that a unit of chromatin is called a chromosome. Therefore, since these chromatids are now individual units, they can be called chromosomes. The chromosomes are moved to the opposite ends or poles of the cell by the shortening spindle fibers. Telophase is the final phase of mitosis. The chromosomes reach opposite poles of the cell, the spindle fibers disappear, and the nuclear membrane reforms. The chromosomes uncoil and return into thread-like mass. In the diagram, you see the beginning of cytokinesis. Cytokinesis is the process of the division of the cell's cytoplasm. This begins during the end of telophase. In animal cells, the cell membrane pinches together, creating cleavage furrow that forms along the equator. The groove or cleavage furrow deepens until the cell membrane separates, forming two daughter cells. DNA replication is the process by which DNA code is copied. It occurs during interphase of the cell cycle, specifically during the S phase. In order for each new cell to function properly, the cell must contain a complete set of genetic material. If the cell is missing a chromosome, it will be unable to create any of the protein encoded on that chromosome. It is also critical that each cell receives an exact copy of the DNA. Imagine a factory in which one worker received a modified blueprint. It would be impossible for them to produce the same goods as the other workers. A similar effect would occur if new cells received different. All right, so why is this important to the growth of organisms? All right, so the importance of the cell cycle to the growth of organisms. Cells limit in size. Okay, cells can only get so big at a time. So here you have 1 millimeter, 2 millimeter, 3 millimeter. Unicellular organisms, cell division is their method of reproduction. Multicellular organisms, cell cycle aids in growth, maintenance, and replacement. Cells only last so long, eventually they're going to die. So if we can't duplicate them and have new cells to replace them, then eventually we would die too. So here we are at the end. I know this is a lot of information and we'll talk about it in class. If you have any questions, write them down. Make sure you have circled your keywords and underlined your main ideas. Please don't write your thought questions unless you really have one until we get to class, because that kind of defeats the purpose of us reviewing our notes to come up with these questions. I'll see you guys in class. Have a great evening.